All right, great. Thanks, Michelle. I'm Sean Jones. I am Process and Development Manager at Arkion Life Sciences. So just briefly my background, so I have a PhD in chemical engineering, but I'm more or less a microbiologist by training. So before joining Arkion, I was more into fermentation, probiotic isolation, and more kind of the molecular biology. And then I joined Arkion more on the antibody side. But as Michelle was mentioning, we really want to look into this idea of specificity within the milk replacer itself. Because IgG is great, but if you don't have the specificity for the antigens required to keep the calf healthy, just having IgG itself isn't necessarily the best idea. So we want to look at kind of the profile of different milk replacers. So we took 5 different milk replacers. So Milk Specialties, DFA, Denkovic, Land O'Lakes, and Provimi. All of these are just off-the-shelf milk replacers that we acquired. And these are, importantly, these are only milk component only milk replacers. So these don't have any plasma in them. They're all milk components, whey components, or other milk-derived components. No plasma are in these milk replacers. And these are the antigens we looked at. So 9 early calfhood antigens from rotavirus, coronavirus, crypto, E. coli, salmonella, and Clostridium perfringens. So looking kind of at all the early calfhood diseases you see. So how we did this was we took the milk replacer itself, we extracted the IgG from the milk replacer, labeled the IgG with a reporter enzyme, this HRP, and then we did the ELISA. So how ELISA works is on the plate itself, we attach the antigen. So let's say it's the bovine rotavirus. We attach that to the plate. We then apply the IgG, which will attach to the rotavirus. The more IgG attaching to that rotavirus, the more reporter enzyme you have, so the brighter the color of the reporter enzyme. And we're looking at the A450 absorbance. So the absorbance at 450 nanometers. That's kind of our raw readout of the assay. So what are the results? So there's a lot on the slide, so we'll kind of walk you through bits and pieces of it. So the sample A through E are listed here. That's not the same order of the milk replacers I listed earlier, so which we randomized it, so try and protect the different companies we're looking at here. On the left-hand side, you see the total IgG measurements of the milk replacers, and in the center there you see the profile of the different antigens. So a few things I want to point out. So first, sample A and sample E, their total IgGs are fairly similar, around 10 milligrams per gram. But if you look at their profiles, they're quite different. Sample E definitely has a higher specificity for those early calfhood antigens. So it's not, the total IgG is very important, but it's also the specificity of the IgG matters as well. You can have a lot of IgG, but again, if it's not going to attach to the rotavirus or coronavirus, it's not doing you a whole lot of good. So you need to look both at the total IgG and the specificity. So you can see kind of here, sample E is clearly has the highest titers overall. A and D are kind of middling, and B and C are on the lower end there. So you're looking at this and probably wondering, can I improve these titers? How do I improve these titers? Obviously, adding more IgG is an option. Obviously, colostrum has lots of IgG. That gets pretty expensive as well. So are there other options besides IgG to improve your titers? Yes. We look actually at IgY. So IgY is the avian equivalent of IgG. It's actually, if you look at evolutionary-wise, they think it's actually a precursor to IgG in mammals. Dr. Saltman's going to go into a bit more of what IgG is and how it works. But it's basically, you can think of this as another IgG equivalent molecule. It's going to bind to the pathogen of interest, neutralize it, and bring it out of the gut. So how do we do this? We did the same thing as we did with the milk replacers. We took our egg powder, extracted the IgY from that, labeled it, and then when we did the ELISA, so now you have one antigen on the plate, we mix the IgY and IgG. So both of them can attach to the antigen and have a greater response in the reading of the A450. So we're reading the combined activity of the IgG and IgY on that one antigen. So what can this do to the titers? So let's look at sample C in this case. Sample C was towards the bottom as is. But now if we dose in just 2 grams of the equivalent of 2 grams of the IgY into a 10-ounce milk replacer dosage, you can see now that you increase the titer significantly. Now sample C is competing more with sample A and D as far as titers. So just by adding a little bit of the IgY, the titers are boosted quite a bit. What about F for sample A now? Sample A was about mid-pack before. Now if you add the IgY, now it's competing more with sample E or out-competing sample E. So really just adding a little bit of this IgY can really boost the titers of the cas-specific antigens. So that was kind of the profiling work itself. I'm going to turn it over now to Dr. Saltman to kind of just describe a bit more what IgY is, how you can use it, and some of the trial results we've seen in some trials we've done with it.
Thank you, Dr. Jones. Appreciate that very much. Just by way of introduction, my name is Roger Saltman. I have spent over 35 years in the animal health industry after several years of being in private veterinary practice. I retired in April of 2019 as Chief Medical Officer at Zoetis and have since been working as a consultant, primarily with small companies to help them to better understand their products, do ongoing research with them, and to ultimately to work on the value proposition that these products have ultimately for the producer. So again, I want to go back to the slide about IgY. IgY, as Dr. Jones said, is the avian version of IgG. They actually look very, very similar. Last year at this meeting, we actually talked a little bit about the slight changes that exist in the protein. These are proteins, and they're part of the immunoglobulin family that Mother Nature has created to ideally tie up antigens of various sorts, typically pathogens, that might invade the body and perhaps cause disease in the animals. The short tails of the Y are designed to be very specific for specific pathogens. So in whatever environment they are in, these immunoglobulins float around, and when they come in contact with very specific antigens, they attach to those antigens, they tie up those antigens, and those antigens are no longer available to cause disease in the animal. The IgY, as Dr. Jones said, is the major antibody produced in chickens and in birds in general. It's found primarily in the animal's blood, but also in egg yolks. And I found it very fascinating because in Mother Nature's wisdom, these egg yolks are very rich in IgY, very similar to in mammals, the richness of immunoglobulins, IgG specifically, in colostrum. So this is Mother Nature's way of assuring that when the chick is born, the chick has access to antibodies until the chick can begin to make antibodies on his or her own. So let's talk a little bit about active immunity and passive immunity. These are terms often used. Active immunity is essentially the immunity that's produced primarily antibodies, but it can be more inclusive than that. These are antibodies that are made by animals when they come in first into contact either with pathogens or some type of antigen. They respond to that interaction with the antigen. Special cells in the animal's body begin to produce these proteins that we call either IgG or IgY, and it ramps up, and there's a lot of this specific immunoglobulin produced, and it helps to tie up the antigen or the pathogen that it's responding to. It does take some time to mount this immune response, and one of the advantages, though, of active immunity is that the animal now has, I'm going to call it the machinery already in place to, when it comes into contact with the antigen or the pathogen again in the future, it can very quickly mount an immune response and ramp up and produce a lot of antibodies. By contrast, passive immunity is immunity that's given to the animal. It can be given to the animal, as an example, for mammals in colostrum. So they passively get a big amount, a very large amount of antibodies in their mother's colostrum. In the case of birds, they're getting a large amount of antibodies through the yolk that was produced by the hen in the case of chickens. Again, the antibodies are already present. They're immediately available, that if they come in contact with those very specific pathogens or antigens that they were produced to be against, they can tie them up. One of the negative things about passive immunity is that you impart this to the animal in various ways, which I'll talk about in a second. The animal has those antibodies available for a while, and then if the antibodies are in the intestinal tract, they are excreted in the manure. If they are in the bloodstream, they're broken down, and again, there's a big shot, and then they begin to diminish. Now, if you look at what's happening in the case of calves, is shortly after they're born, they get a large amount of colostrum from their dam, and the passive immunity in that colostrum provides a lot of high level of immunity to that calf in the very first few days after it's born. But then that immunity begins to wane, both in the intestinal tract and also in the system itself, in the animal system itself. And then there's a period of time where the animal is being exposed to pathogens, and the animal now has to mount its own active immunity. As I've already mentioned, that takes time. And during that time is when animals can get sick. So that first couple of weeks, anyone who's raised calves knows that's the critical period where calves can break, in particular with scours. So the Arkion product, which we call EggTek-C, is produced. Actually, the EgCel is the platform that I'm going to describe on how passive immunity is created in egg powder. So very specifically, bacteria, viruses, and some other antigens are injected into a hen. The hen now mounts an active response, produces immunity to these organisms, and she excretes some of this immunity into the yolk of her egg. Okay, so that shows you in the picture the little Y shapes in the yolk of the egg. So again, preparing ultimately for a chick to be born to have this immunity given to the chick. In the case of production of EggTek, the eggs are harvested, they're cracked open, the entire egg contents, both the yolk and the white of the egg, is dried down, and a whole egg powder is produced through spray drying. In some forms of this kind of approach, they may just use the yolk because that's the richest part in terms of antibodies. There are other types of products where the antibodies are harvested and purified into some type of solution, and even liquid egg can be used for this purpose. Again, in the case of the EgCel platform that Arkion's created, this is all made into a whole egg powder. The way that IgY works in terms of protecting calves is that the powder is mixed in with milk replacer or whole milk and fed to the calf. These antibodies then are introduced into the intestinal tract of the calf. The antibodies stay in the tract of the calf. And if there are either bacteria or viruses that the antibodies come in contact with, they tie up those pathogens so the pathogens can no longer do any harm to the animal, and they are excreted in the manure. So this is the way that EggTek-C actually helps to prevent scours or diarrhea in baby calves. I might point out that the antibodies stay in the intestinal tract. They do not go through the intestinal tract into the bloodstream. They actually never enter the animal herself, so there's no additional systemic protection that's created. Now we've done a number of studies to look at how the EggTek-C binds with different pathogens. What you're looking at here are two pictures. In the case of the picture on the left is a photomicrogram of E. coli K88 with K88 antigen on it that has been exposed to milk replacer with just standard egg powder introduced. Not much color there. But on the right is E. coli K88 antigen with EggTek-C that's been introduced into the milk replacer. The photomicrograph is such that the more binding that exists, the brighter green is the fluorescence. So you can see that the antibodies have actually opsonized or attached to the E. coli, producing all these very bright fluorescent green dots. So it actually binds to in this case, to the E. coli and prevents the E. coli from doing damage to the calf. We've done other studies that we're in the process of summarizing and hope to place into the scientific literature, very specifically with both bull calves and also with heifer calves. Typically, the bull calves are often colostrum deprived, so when they're in their first few days of life, they're exposed without any protection at all to some of these pathogens, and they can get very sick, have really severe scours from that. In the studies that we've done on over 400 calves, we find that the use of the EggTek-C reduces mortality significantly, and particularly in calves that have received no colostrum. So they have no protection. When we introduce protection through the EggTek, there's significant reduction of disease. In the case of a heifer, in a trial done in a large dairy, on almost 300 heifer calves, very well managed dairy, where colostrum is not an issue, the calves all get colostrum. We found that, particularly in the face of some challenge, as an example, in the time of the year that we're coming into, that the addition of EggTek can significantly reduce both the longevity of scour events that occur and also reduce the severity of scours if an animal gets scours. We also found that fewer treatments are needed when animals have been fed EggTek every day in their milk replacer. We know that as well that challenge can be different at different times of the year and in different calf ranches, different dairies. And we know that in the face of severe challenge, a higher level of EggTek is required than in the case of more minimal challenge. So let me close by again repeating that the significant technology here is called the EgCel technology that Arkion is using, and from that different products are being produced. There's the EggTek-C for calves, EggTek-S for swine, Active Egg, which is very specific for companion animals, and there's even actually a human product that can be used to help prevent disease in humans. So with that, I'm sure that Dr. Jones is available, as am I, to answer any questions that you might have. Yes, sir.
You talked about the IgY being able to, not gene, anywhere, bind to the antigens.
That's right.
Is it recognized by mammalian immune system, like neutrophils bind to it, will get cytokine production and all that?
It's great. It's a really great question. So I've really just been talking about the antibody response as part of the immune response. But to your point, there are other elements in an immune response. They're not affected by this at all. But what's happening, if you want to think about it this way, is in the case of the IgY, which is the avian product the avian antibody that's first opsonizing those pathogens. The pathogens are also present and are being responded to by the calf herself, and so she begins to make active immunity herself. But there's the period, it takes a while for her to establish that a high level on her own. So what this is doing is bridging that gap. So all the elements that you're talking about in the immune response, they also are present. So the calf is mounting her own immune response, but it's a muted response. We know that animals, when they're young, they're still developing their immune systems. So this is, if you will, is an aid to help her to bridge the gap till she can mount a sufficient immune response to protect herself. Does that make sense to you?
What I'm getting at is, will the IgY help stimulate that innate immunity?
Do you want to speak to that,
Yeah.
John?
Yeah, so the IgY should not, it should not recruit the native immune system. So the IgY, it stays in the gut. It's going to bind to the E. coli or the rotavirus, and it's going to then be excreted from the gut. But it's not going to activate the calf's own immune system. As Roger said, so what happens is, so let's say the calf sees E. coli. The IgY will bind to the E. coli, get rid of it. Some of the E. coli will still be there. So as Dr. Saltman mentioned, the own calf's immune system will activate to that. And so what the IgY is doing is decreasing the pathogen load. So that the calf will still see the pathogen and still mount its own response, but the IgY is not initiating the response. It's just getting rid of the pathogen, but the calf will still see the pathogen and recognize it and begin mounting its own response. The IgY is kind of helping get rid of more of it so it doesn't get as sick.
Mitigating the level of infection.
Correct.
That's why we tend to see scours primarily in young calves, as opposed to, say, heifers who are 3 or 4 or 5 months of age. They now have developed their own immune systems that, as they get exposed to those same pathogens, they're able to fight them off themselves. Yes, ma'am. Could you repeat your question? I'm not sure I heard you very well.
With consistent use, is there any concern of pathogenic resistance to the product?
In short, no. Generally speaking, antibodies are very specific. They're the interaction. Think of it as a key and a lock, and so once the right key finds the right lock, it locks up and they stay together. It's called opsonization, and it's and again it is having no effect, as Dr. Jones has said, on the animal's own ability to continue to develop its own ability to make immunity.
It's not acting similar.
No, it's not like an antibiotic in developing resistance.
And in the generation of it, so the IgY is polyclonal. So we introduce the pathogen or the antigen to the bird. The bird then selects which regions on there to develop the antibodies from. So from batch to batch, they may be selecting different parts of the rotavirus to attach to. So we're not always targeting just one part of it. We give it the whole organism to figure out where best to attach to. So there's always slightly different from every bird.
To my analogy, there's multiple locks on every organism. Yes, sir.
Just like add a comment, because really just sort of kind of prevent attachment of the pathogen to the epithelial cells. So it doesn't do anything to the bacteria per se, like antibiotics. So we're not releasing any, change in the bacteria cell. Okay. So we're not releasing any immune response from the cows, from our cow. Okay. So it's different. It's just, I think it's more like a physical balance.
That's right. It's mechanical.
So now comes two questions. How much you have to feed this thing to the animals? With all the proteins, IgY, IgG, the protein amino acids, so how are we going to prevent it being digested in the stomach, right? So you have to stay in GI tract to make it functional.
Right.
That's my first
Yeah,
question.
sure.
And the second question is, the late professor at Poultry Science at the UW, Matt Cook, I think he licensed this kind of things, the feed IGY to prevent animal diseases, whether it's chicken, pig, or
He ma-
cattle, I don't know. Is these things that the patent, whatever, expired, or you actually get a license from it?
So it's a really good question. I'll answer the second one first, maybe. This is not the notion of harvesting IGY from chicken eggs is not a new notion. It's actually been, it's like we're talking at least 30 years in the scientific literature. There are different techniques that are used. So maybe what you're referring to, perhaps what he licensed or patented, was a specific technique, but there are others. Yeah.
Yeah, so he has specific antigens patented, but the whole concept is, as Dr. Saltman said, it's been well known and yes.
It's so there's some specific patentable elements of it, but the general concept is not new and not limited.
There was like a company called EggTek in medicine. I couldn't find that relevant information anymore. I just did a little research on my phone.
And the literature has lots and lots of references to different companies, actually, and not just limited to the U.S. Certainly in Asia, there's a lot of this work that's done. Your second question is a really important question, and it's the question of being able to pass through, and at that stage. So it's very quick pass through, and remember that even though we think of these as ruminants, at that stage they're really simple stomached animals, and so the passage rates are quite high of milk, and so the milk begins to get digested in the stomach, but of course a lot of it is in the small intestine, and that's true in general of proteins moving through the stomach. So we would expect, I certainly would imagine that there would be some degradation in the pass through from the stomach, but practically speaking, most of it is still getting to the lower tract in the small intestine. It's a really good question, though. Yeah.
So may follow that a little bit. So is AGY more resistant to break down whatever by enzyme, by acid?
So yeah, there is breakdown of IgA is the most common antibody in the mucosal linings. It's true in GI tract. It's true in the nasal passages, but yes, like everything, it's not forever. There is breakdown over time. So there has to be. So the ability to actively create new immunity is ultimately critical, right, long term. So yes, but it is broken down, and it's in this case though the passage rates are probably more of the, as an example, IgY that's introduced into the intestinal tract. More of it probably is passing out in the manure than actually is being broken down, and which is again why the recommendation is that it should be used twice a day, at least twice a day. So it's put into milk or milk replacer twice a day. And the other part of your question is also important. We tend to think of this as an all or nothing type of response, but in fact, the level of challenge, the number of bacteria or viruses that an animal is exposed to is not static. So in certain times of the year, it'll be a higher level of challenge. If management on the farm is such that, let's say that there's more animals that are calving through a small pen, and so the level of challenge that gets to calves is higher. In those situations, we likely need higher levels of IgY to be protective, and that's what we saw in our studies, that for calves, bull calves, that are colostrum deprived in particular, and who are exposed to high levels of pathogens, that they particularly it particularly benefited by the addition of the IgY to help lower the level of the pathogens in their gut.
The load, just like he said, reduce the load. So
That's right.
Maybe you needed 100 bacteria to cause infection, but you only have 10. Then you don't have infection happens, but the bacteria is there. Then the animal have enough time to develop their own immunity. Right?
Exactly. And just for your knowledge, we've looked at different doses from 4 grams all the way out to 20 grams per day, split into 2 feedings. And certainly the higher doses were particularly beneficial for the bull calves. Are there any other questions? Yes, sir.
Your slides indicated that this type of product has been used in a number of species.
Mm-hmm.
Has there been any reaction from the host species to the IgY as a foreign protein?
Well, remember, this is being given orally, so that the thing I always try to remind people is from the mouth to the anus, that's all outside the body. So, it's not in the body, actually.
Why do we have food allergies?
It's a really and that's it's a good point, actually. It's a reaction to at the mucosal lining, but we've not, to the best of my knowledge, we've never seen that with IgY. But it's a very good point.
Thank you.
Sure. Yes, ma'am.
Couple questions. How much does it cost? So I would assume you're recommending 2 grams twice a day. Is that right?
It's sort of as a maintenance level in low challenge situations. The answer is yes. In high challenge situations, where there's a high incidence of disease or expected, then we're recommending higher levels in the range of anywhere from 5 to 10 grams twice a day.
So your maintenance level is the cost per animal.
This is
It's about 3 cents per gram list price. So that would be about 13 cents per day.
That's pretty cheap.
That's the list
Well,
price.
but
Yeah.
it's maintenance fees too.
Yeah.
And once you get to challenge, you're probably
Yeah. Oh, yeah. Yeah. Of course, the thing is, what is a case of scours, particularly severe scours, cost? It both in terms of the risk of even death for the animal, but certainly the animal gets way behind in terms of her growth curve. It seems to me anyway to be one of the most cost-effective strategies for prevention of scours that we have. Okay, go for it.
Second question, what are all the antibodies that are in the product that are common?
Yeah, was that in one of the slides? I think.
I must have missed it.
So
Yeah.
so the 9 antigens we looked at, so the rotavirus, all 9 of them are right.
Yeah.
I'll think of it now, because I have another question I need to ask.
These are all good questions. Thank you for asking them. From the other member? Yes, ma'am.
Is there any correlation at all between the use of this product and eventual ruminal activity, or does it promote that?
I wouldn't think so. I wouldn't expect it to. I'll put it that way. We haven't looked at that specifically, but it really I can't even imagine how they might be related. So much of the development of the rumen is a function of solid feed that calves get on, particularly forages, and so that stimulates them.
The development of the microbial population.
That's right. Which is happening in the rumen, right? Yep. You're right. You're absolutely right. But I wouldn't expect it to have any impact.
And the only thing it would do is help reduce the E. coli or Clostridium perfringens developing in there. It wouldn't necessarily prevent any good ruminant bacteria from growth. That's outside of the spectrum that we introduce.
So, yeah, so these hens have been immunized very specifically with calf pathogens, not any other types of organisms. Right? Yes, sir.
So this might be away from cattle. So you didn't mention you have EggTek-C for the swine.
Yes.
So you have commercial products for the TED yet?
Yes.
Right?
Yes.
Other products?
No.
Or any research going in that direction?
I'm sorry, I missed that. PEDV for swine?
Yeah.
Oh yes, so our swine product has PEDV in there.
So you do actually have a commercial product.
We have a swine product for mash feeds, yes. So if you have a,
Does it have this egg yolk powder added to the mash feed?
yes.
So that for the cattle you add it to the milk, right? Replacement.
For the calves we put it in the milk replacer.
Yeah.
For the swine it would be in the mash diet.
But I would be quick to add that there's no reason why it could not also be included in a starter grain for calves.
Yeah, I think so because I was doing cloning work. Okay, everything comes so nice, and then 3 days later we have this uncontrollable PD. You just see the piglets die from dehydration. It's a severe diarrhea.
Yeah, that.
If we had something that would help, then I would save lots of my cloned piglets.
Yeah, unfortunately, because of the way that piglets, nurse,
Right.
there's no way of introducing it. Yeah. Yeah. Thanks.
So I did remember my question.
Okay.
So maybe more hypothetical than anything. Would you expect an IgY for coronavirus to perform differently than IgG coronavirus in the animal?
Say it again, sorry.
So say you've got an IgY that's for coronavirus specific antigen. Would it perform differently than an IgG virus that is needed to make proteins or something like that?
So we don't have any data to answer that either way. There is one paper, I think from '97. They took an IgG and an IgY. They found that in vitro they had the same neutralizing titer for, I think it actually was coronavirus. And then when they fed to challenge calves with coronavirus, the control, completely all the calves died in the control. And they found at what dosage you had almost complete survival of the population. And they found that the IgY needed less total titer than the IgG. And they speculated on that. It's been the one paper, so I don't know. I would expect them to be similar, but there may be differences, and we haven't really teased all of that out yet.
Good question.
The other comment I had about the starter feed, I don't know that your IgY would survive quality.
It's a really good question. We actually know that it can. Yeah.
At certain temperatures, it can.
What's the maximum temperature?
I would say about 140 Fahrenheit would be as high as we could go with the good survival.
A lot of plants will be higher.
And we are working for different formulations, but yeah, as in the current product form, I would say 140 Fahrenheit is the maximum, but yeah.
You're right, though, when in some pelletizing plants, the temperature is higher than that. So, but under limited heat conditions, it can remain in the pellet. Otherwise, there'd be no way to provide it to swine, right? Any other questions? Thanks to all four of you. You presented us with some great questions.
We have 3 against the 4.
Thank you.
Yeah.